Okay, I guess it's time to go ahead and start and finish this week. So we left off just getting out of bacterial viruses. Thank God, right? So now we're talking about viruses that infect humans. And so we'll start off talking about poliovirus, but I want to talk a little bit about the disease first, because really my interest is disease. That's why I became a microbiologist. So poliomyelitis is a disease that probably none of you have ever had to deal with at your age. Even at my age, it was, when I was younger, it was a relatively rare disease, but it's still a very severe disease, and it's caused by a group of viruses that we're talking about now called the picornaviruses. This is a really great name that virologists have given to this class or this family because the name actually has a lot of information in it. Pico meaning small, or pico, if you want to pronounce it that way, and RNA meaning that this is an RNA virus. So that name just kind of gives away some of the information about it. But it can actually, unfortunately, at some frequency infect the central nervous system. And that's when the real problem begins, because the disease, very unfortunately, it's unfortunate if it affects anyone, but it's always more tragic, right, when it infects children. And so it's predominantly seen in children under the age of 5, but certainly adults can be infected as well. Certainly our President Franklin Delano Roosevelt was infected as an adult. But most people who get it under the age of 5. And most people who become infected might, or some of them at least, might not even know that they're infected. If they do know that they're infected, it's usually just a little bit of fever, some tiredness for a little while, probably some diarrhea, but then the infection clears. That's, from the virus's point of view, that's the preferred outcome. There is no reason that the virus needs to spread and infect the central nervous system. But on occasion that it does, luckily not more frequently than it already does, but about 1 out of 200 cases of poliovirus infection of a human will lead to AFP, or acute flaccid paralysis. It leads to irreversible paralysis, usually in the legs. This recovers to some degree, but you can oftentimes see pictures, and I'll actually show you an Egyptian stele in just a second that has the classic characteristic withered leg. The more severe form, which is, or was thankfully much more rare, is bulbar polio. So this paralysis actually affects the muscles that allow inspiration, so breathing. So people would actually die from this quite frequently. As a matter of fact, people had to live in iron lungs to help them breathe. Now, polio, or poliomyelitis, is actually a very ancient disease. It's actually, this is an Egyptian stele that actually portrays somebody with the characteristic withered leg. So it was a well-known disease even, thousands of years ago. But that's very different. There were no historical documented epidemics of polio. It was described in the historic literature as a sporadic disease. It still had the same paralysis and same fatality associated with it, but there weren't these major waves or epidemics. We really only saw epidemic polio within about the past 200 years. So a little bit before my time, in the 1950s and in the 1940s in the US, there used to be, almost always in the summertime, epidemics where as many as 20,000 young people would be infected with polio, many of which would develop paralysis, and some would actually die. It was a very terrifying time for parents back then because there was no vaccine and people really didn't understand the disease very well. So children during these epidemics wouldn't be allowed to go to the local swimming pool or to the local swimming hole. Because people did realize that it was being spread probably by fecally contaminated water, but there was no vaccine for it. But it's really fascinating that we only saw epidemic polio within about the last 200 years. And then the other fascinating thing about it is that poliomyelitis this epidemic form predominates in countries with the highest hygiene standards, which is the opposite of what people would expect. Epidemiologically, you think of diseases that are spread by fecal contamination of water as things that are going to happen in areas that don't have good infrastructure, don't have water treatment plants. But in fact, people in the underdeveloped world at the time never really had to recognize or never had to deal with these epidemics of polio. But in the summertime, there were huge numbers of children who were dying. And so those that did survive would survive because they would be able to live in these iron lungs for a while, which would help them breathe. So negative pressure inside here would actually cause the chest cavity to expand and to draw in air. Now these children would oftentimes recover to some degree, so this wasn't necessarily, people in iron lungs weren't necessarily going to die, but there would be entire wings of hospitals dedicated to people with this bulbar form of poliomyelitis. Before I leave this, I want to tell you, it's a really interesting epidemiological story as to why developed areas like Europe and North America in the last 100 years had these epidemics. It actually has to do with our very hygienic lifestyle. So as it turns out, in areas where polio didn't occur epidemically, what would happen is that as a mother gave birth to a child, if that mother survived to the age of having given birth, chances are she was infected as a child with polio. And like most viral diseases, if you survive the infection, you become immune for life. So what happens when an immune mother gives birth to a child, there are certain antibodies that cross the placental barrier. So an antibody class called IgM. I know you haven't taken, maybe some of you haven't taken immunology. Some maternal antibodies cross the placental barrier. And so when the child is born, the child is passively immune to some of the same infectious agents that the mother is. And then, of course, there is that time during breastfeeding where the child is now getting antibodies that the mother has through the milk, especially through the colostrum, the first milk that's produced after birth. So these children now actually have within their blood system a series of antibodies that protect their central nervous system from the virus. So they have maternal antibodies circulating in their blood so that if they are infected with poliovirus, and they will be in these environments, so that when they're in an environment where fecal contamination of water is quite common, and so the poliovirus is there, these children are infected, their intestinal tracts are infected during the time when their blood system actually has antibodies from the mother. Because what happens in a virus such as polio, the normal case is that the virus now infects the pharynx or infects the intestinal tract and is excreted in the feces. And what allows for this infection of the central nervous system is this, what's referred to as a viremia. So the virus at some frequency gets access to the bloodstream. So when viruses are being circulated in the bloodstream, that condition is called viremia. If these were bacteria that were in the bloodstream, it would be called bacteremia. In general, this would be referred to as septicemia, so an infection of the blood. But this viremia, in the case of poliovirus, would allow a situation where the central nervous system could potentially be infected. But if the baby has antibodies from mom, the virus cannot infect cannot infect the central nervous system. It can infect and does infect the intestinal tract. And that allows the baby to limit the infection to the intestinal tract. And now that child will be immune to the virus on its own for the rest of its life. So these children develop the intestinal form of polio, but they never develop, or they seldom develop, the more serious forms, the more fatal forms of central nervous system. In the US, these children might still be born to mothers who are immune, they have antibodies, but the babies wouldn't be exposed to the virus initially because of the high hygienic standards, because our water system is cleaner and the virus is much less frequently found in the water. So what happens over time is that children stop breastfeeding, so they're not getting antibodies from breast milk anymore. The antibodies that were passed via the placenta into the bloodstream of the baby break down over time. It's a temporary passive immunity. That leaves the child completely naive in terms of poliovirus infection. So now, when that child at 4 or 5 or 6 years old or a teenager gets infected with polio, if the child is lucky, it's just infected in its intestinal tract. But that transient viremia that occurs can actually introduce the virus now, because there are no antibodies to neutralize that virus. That virus can now infect either the meninges or the brain. So the meninges are the protective tissue that surround our central nervous system. And so the infection of the central nervous system can lead to the paralysis. So that's the reason why. So it's almost counterintuitive. You expect that infectious diseases are going to be much more predominant and much more severe in less developed areas of the world. But this is an interesting example of how that's not always the case. So as I mentioned, in the 50s, there really wasn't a vaccine. The vaccines there were a lot of people developing them. The first vaccine that was actually deemed safe and acceptable was generated by Jonas Salk, who later won the Nobel Prize for this. This was a virus that was injected, so it's a parenteral vaccine, and it's an inactivated virus. So this was really a very simple vaccine. What Salk did was basically to take advantage of the fact that somebody whose name escapes me right now had come up with a way to grow African green monkey kidney cells, called vero cells, in culture. So growing tissue cells at this time was pretty difficult. So they were able to actually grow the virus now in the laboratory to high titers or high amounts. And then basically all Salk did was to take that virus and inactivate it. He treated it with formalin or formaldehyde, which denatured the proteins a little bit, made it so that it couldn't cause an infection, so it was completely safe to inject into a child, and then that child would become immune, or certainly much less likely to be inactivated. So the Salk vaccine worked very well because it induced a class of antibodies called IgG. So this is the form of antibody that predominates within the plasma of our blood. If you think about, as I just mentioned, what causes or what allows the poliovirus to cause infection of the central nervous system is the fact that it has, at some point during its life cycle, a viremic stage. If there are antibodies against the poliovirus in the form of IgG circulating in the plasma, in the blood, then that virus becomes inactivated. And so it's not capable to infect the central nervous system. So the Salk vaccine was very, very effective. So if you look in this graph at the number of cases of poliovirus over time, you can see that here at this era, this is the introduction of the inactivated vaccine invented by Jonas Salk. And you can see this tremendous crash in the number of cases. So it was very, very effective, but not entirely. It didn't really get rid of poliomyelitis. So it induces this serum IgG to prevent viremia. So it's very good at preventing it's very good at preventing the disease poliomyelitis. But it's problematic because it required boosting. And this was a muscular injection. And so if any of you who have had injections directly into the muscle, these are painful. And when you're a small child, they seem even more painful, right? We still probably all have some fear of needles. So parents getting their children to the pediatrician to get this multiple rounds of injections was not easy. So this was a problem with it. It required boosting. The other problem was that it didn't elicit the mucosal form of antibodies called immunoglobulin A, or IgA. I'll show you a picture of these in a little bit. Because we have a different class of antibody that our bodies make to bathe the mucosal surfaces of our bodies. So our respiratory tract, our urogenital tract, our gastrointestinal tract, our conjunctiva, have antibodies, a specific class of antibodies called IgA secreted onto the surface with the idea that they bind to bacteria, specific bacteria or viruses or parasites to inactivate them before the infection occurs. So the vaccine that Salk came up with didn't elicit this mucosal antibody response. And so people could still be infected with polio. They just didn't develop the central nervous system complications. So the virus was still going to circulate within the population because people could still be infected. They would get gastroenteritis and diarrhea, and they would pass the virus on, and it could potentially infect other people. So this is a great virus for preventing central nervous system complications, but it didn't do anything to help us get rid of the polio virus. So the next vaccine that came out, and these two men were competitors, Albert Sabin came out with a vaccine that was, in my estimation, a little bit more clever, and it's actually more effective at getting rid of the virus from the world, helping to make it extinct. This was an attenuated live virus. So what Sabin did was simply by taking a wild-type virulent form of poliovirus and growing it in tissue culture cells over and over and over again, it acquired mutations randomly that made it less well adapted to infect people. It could still infect people, but it couldn't infect the central nervous system as well. Didn't lose that completely. So this is what's meant by attenuated. Its virulence was lost just by serial passage. Actually, I think he passed it, somebody came up with a vaccine passing it through mice brains. I don't know whether that was Sabin or not. But in any case, the virus just acquired mutations that at the time they didn't know what the mutations were, but they knew that it was now no longer or less likely to cause disease. But since this was a live virus, you could infect a child in vaccination. So this is the oral form. And what happens is that the child's pharynx and intestinal tract becomes infected. The child makes its own immune response, which includes serum IgG as well as mucosal IgA. So that if the child ever becomes now exposed to the wild type, the virulent form of the virus, the child is now immune to actually even becoming infected. So that's part of the reason why it's the Sabin vaccine, this oral attenuated vaccine, which is the basis for why we can and will hopefully shortly eradicate poliovirus from the world. We can make this extinct. This is one of the few infectious agents that we have the possibility of actually driving to extinction, in part because poliovirus infects no other animal on Earth except humans. If we can make enough people on Earth, humans on Earth, immune to this virus, there's no place for it to hide. There's no animal reservoir where it can jump back into the human host again. So this is an unusual situation. Most bacteria or viruses have some kind of another reservoir in the environment. We have eliminated smallpox virus, and smallpox virus is the same as polio in this regard. It only infects humans. So when enough humans became immune to smallpox, it couldn't spread any longer, and there was no place for it to remain in the human population. So the Sabin vaccine is really critical to that role, or to that possibility of making poliovirus extinct, because it generates IgG and IgA. It's less safe, though. So like anything else, it has its problems. When used in the U.S., it actually resulted in about 10 cases every year of paralytic poliomyelitis because of the virus. So remember, this is a live and attenuated virus, so it has acquired, at that time, uncharacterized mutations that made it less likely to cause polio. But those viruses, at some frequency, had the ability to revert. So those mutations, which made it less virulent, could now be lost. And so the person that's actually infected, that took the sugar cube or the drops in the mouth, that virus could mutate and cause polio in that person. very few cases. And so when the cases in the US were in the thousands per year, this was an acceptable risk, right? You were willing to risk 10 cases of vaccine-derived polio to prevent perhaps hundreds or thousands of cases of polio. Eventually, we reached a point where there were so few cases. Actually, the last case of polio, wild-type polio in the US was, I think, in 1994, and in the Americas in general around 1998. So in the year 2000, the US stopped using the Salk vaccine. We don't vaccinate with it any longer because of that risk. Anybody who needs to travel to an area that is endemic for polio, and luckily there's only 2 places left, Afghanistan, so soldiers that go there are vaccinated, and Pakistan still has this. So those people going to those areas will be vaccinated with the Salk vaccine. So we've gone back to the Salk vaccine. But the Sabin vaccine is still being used elsewhere, especially in Afghanistan and in Pakistan. I'm having a hard time coming up with words this week. I guess I'm still tired. So before we leave this, I just wanted to show you a representation for those of you who haven't had immunology yet. So this is what serum IgG looks like. So this is the antibody, immunoglobulin G, that has antibody binding sites here and here, but it's a single chain. So it has the ability to bind to 2 identical epitopes, sometimes usually protein perhaps, but also polysaccharide or lipid. This would be the secreted form. This is the form of an antibody that's secreted across our mucosal surfaces to coat the surfaces. And so this is a dimeric molecule, so it has 4 antibody binding sites. Is there something else I wanted to say about this? Actually, I'm just going to go a little bit through the history of these vaccines a little bit, and then I want to talk a little bit about some of the current concerns that go on with vaccines for this. So the early vaccine trials for polio in the 50s were freaking disasters. I mean, people were getting sick. People were doing some of the most unethical things you could imagine. One investigator actually was inoculating patients in a New York City mental hospital. These are things that could never happen today, thankfully, and in part because of some of the really unethical things that some of these people did. So these things were front page stories. And so parents, anytime somebody like Salk came up with a viral vaccine that was potentially good, it was really difficult to convince people to allow health officials to vaccinate their children. So Salk actually had to first inoculate himself, his wife, and his sons in order to gain some confidence that this vaccine was potentially safe, at least. So the vaccine trial was a big deal. It was a huge vaccine trial, and it was actually in 1955. It wasn't until then that it was determined to be safe and effective. The vaccine works, it's safe, effective, and potent was one newspaper's headline. The study group was huge. So it was nearly 2 million children in second grade. They were referred to as the polio pioneers. And so here's the ethical dilemma that some people had about this at the time, and rightly so. Of those nearly 200 million children, nearly 750,000 were received a placebo. So they had no idea. They thought they and their parents thought they were receiving a perfectly, potentially life-saving vaccine, but in fact they were getting sterile saline injected. The nurses or doctors didn't know which needles contained placebo and which ones contained the vaccine. Yet 3/4 of these children were going to be left without protection. So here's a portion of the results from one portion of the study. So here's the treatment group of 200,000 children, and here's the control group. So if you look at the number of poliomyelitis cases, paralytic poliomyelitis cases that come up in the children that got the placebo, 71 children got it, compared to only 28 children that out of 100,000 that got it if they got the actual vaccine. So this is the ethical portion. It's like many of these children that got the paralytic form might have been spared that form if they actually had not received the placebo instead of the actual virus. And then this was the group that were followed whose parents failed to give consent. So 46 per 100,000. So the vaccine was done in this double-blind, randomized, from an epidemiological perspective, this is referred to as the gold standard. So the experiment relied on volunteer subjects who were randomly assigned to either the treatment or the placebo groups. So there were people, including Jonas Salk himself, who came up with this vaccine, who had objections to this. And here's a quote from Salk that I found. "A beautiful experiment over which the epidemiologists could become quite ecstatic, but would make the humanitarians shudder." I like this quote and I don't like this quote because I have friends who are epidemiologists who would have had problems with this too. It's not to say that epidemiologists don't care. They do. But so there was a lot of controversy about this. Now, I mentioned that this virus is potentially becoming extinct. So here is polio in 1988. I know none of you, except maybe one of you, were born in 1988. But while there wasn't in the U.S., there was still in our hemisphere, there was polio, and it was widespread, even still throughout Europe. Now in 2016, I wish it was still last century, there is really just in Afghanistan, in Pakistan, and in Afghanistan. And the problem that that people are running across now is twofold. We may be coming up against a real problem with trying to spread vaccine in Afghanistan and in Pakistan for two reasons. One is now there is a widespread belief and distrust in Western medicine that this is actually, there are rumors that there are products in the vaccine that make men sterile or impotent, and that it's the West's idea to against Islam. The other, and I think this is a bigger problem, is that the U.S. actually did this to ourselves. So the U.S. Central Intelligence Agency set up in the hunt for Osama bin Laden this sham vaccine program in Pakistan, where healthcare workers were going around not only giving the polio virus, but they were all vaccine, but they were also giving vaccines against hepatitis. That's an injectable vaccine. What they were doing, in fact, was not only, they were in fact giving legitimate vaccines. The vaccine workers didn't know what they were doing. And they were actually giving legitimate vaccines, but the needles were being collected for DNA testing. They had the Central Intelligence Agency had DNA from one of Osama bin Laden's sons who lived in New York City. And so they used the DNA samples that were collected from those needles in order to find a concentration of people most closely related to Osama bin Laden. That's how they located where he was. So once that news came out after bin Laden was killed, of course, there's this tremendous backlash now against vaccine workers. Vaccine workers have actually been killed. Polio virus vaccine workers have been killed since that time in retribution for what the CIA did in setting this system up. So you can see how complicated these vaccine efforts become because politics ends up mixing in with science and with really tragic outcomes. So while we have the possibility to potentially eradicate one of the worst infectious diseases the world has ever known, we may not actually be able to do it. It may be able to persist in this area because of these things. And vaccines have done such wonderful things. I mean, I'm not going to get up on the soapbox too much, but I mean, if you look at things like diphtheria, for instance, there are no cases of diphtheria in the U.S. now. It's been 100% eliminated. And you can go through the whole list of measles, mumps, pertussis. But this fear of vaccines is a tremendous stumbling block that public health care workers. So I know many of you are interested in public health. So these are things that you need to be aware of. Okay, going to get off of my political, semi-scientific kind of lecture thing, and we'll actually now talk about poliovirus as a virus and how it works. So it's a really simple virus, and it's very small. Remember that the coronaviruses are very tiny. That name tells you. They're not much bigger than a ribosome. But within that ribosome-sized particle, or slightly larger, lies an RNA genome with the capability of causing paralytic poliomyelitis. The icosahedral head of a poliovirus is made up of only three proteins, BP1, 2, and 3, and BP just standing very logically for viral protein 1, viral protein 2, and 3. And so they're put together into these vertices and these faces of the icosahedron. The way the virus infects a pharyngeal cell, or hopefully not a cell of the central nervous system, is that it binds via two of those viral proteins, 2 and 3, which lie at the vertex of one of the capsid pentamers, to a surface receptor, a protein, glycoprotein on the surface of the pharyngeal cells or on the cells of the central nervous system, which will then trigger endocytosis. So it's a kind of a receptor-mediated endocytosis. The virus is taken up into that endosome, and then the endosome changes its chemistry, probably by acidification. But what that does is that changes the protein conformation of viral protein 1 within. This only occurs within the endosome, probably due to acidification. But now a pore will actually form, so that this single-stranded RNA genome of the poliovirus is now transported from inside the nucleocapsid and released freely into the cytoplasm of this cell that's just been infected. So this is a process referred to as uncoating, because the virus is now coming out of the coat. So this genome, the poliovirus genome, is very similar to virtually all of the coronaviruses. So this same situation is occurring with rhinoviruses that cause most of the common colds. This plus-stranded, single-strand RNA genome, meaning it's the sense strand. So this piece of genome is actually the same as a piece of messenger RNA. But it has on one end a viral protein G, or VPG. This is a characteristic of all members of the coronaviruses, but we're going to talk about it in the context of poliovirus. So that genome, as it comes into the cell, is not 5' methylguanosine capped. So this genome shown here is in the same sense as messenger RNA, and it can and will be translated by eukaryotic ribosomes immediately upon infection, despite the fact that it doesn't have the characteristic 5' methylguanosine cap that's required by ribosomes to recognize messenger RNA. When you and I make messenger RNA and transport it from the nucleus into the cytoplasm, it's that 5' methylguanosine cap that allows ribosomes to bind on, find the start codon, and start translating. This RNA doesn't have it. It has a way to trick, in essence, to fool a ribosome. These are called iris sites, or internal ribosome entry sites. It's a complicated secondary structure to this messenger RNA that is sufficiently confusing to a ribosome that the ribosome just says, oh, the heck with it, I'm just going to translate it. It doesn't really say that. But it's somehow that complicated structure allows the ribosome to bind to that site. So that this genome that comes in, here's the 5' end with that viral protein G attached to it. On the 3' end, it has a polyadenosine tail, just like yours and my messenger RNA. That's translated by a ribosome. And this is one gigantic gene. There is no stop codon in here. It makes what's referred to as a polyprotein, about 2,200 amino acids in length. So it's a gigantic protein made from the entire genome. So it's monocistronic, but actually, in the end, it actually expresses 20 different proteins with different functions. Some are structural, some are enzymatic. But this polyprotein, as you see it here, actually has a autoproteolytic activity. It has the ability to digest itself, probably in trans. So probably a second protein made, a second copy of this polyprotein, will now cleave itself here and here. And then the proteolytic activity changes because now the proteolytic activity probably resides in a central portion of this protein. Once that portion is freed up, it can now recognize other areas of itself and cleave that protein in waves until actually the protein is digested into all of the necessary 20 viral proteins. But the other thing that's really important about this is that one of the proteins that's cleaved off from this, so in this cartoon it's shown as this carboxy terminal region, which is cleaved off. This is the RNA polymerase. This actually probably I should change this title to viral replicase, because this RNA polymerase is an RNA polymerase. It's going to make RNA, but it's going to make an RNA copy of an RNA template. So that's by definition viral replicase. So what happens shortly after this protein is translated and proteolytically cleaved, this viral replicase is going to make a copy of the genome that came in. When you make a copy of a positive strand RNA, that copy is by definition a negative strand. That's what's shown here in this slightly yellower section. It still has a viral protein G attached to its 5' end, but that is what's known as the antigenome because it's antisense to the genome. So what has happened is that viral replicase has used this VPG protein as a primer. So this is a very unusual situation as well. So you realize that, for instance, that DNA-dependent DNA polymerase that you've all talked about in terms of replicating DNA requires a primer. This one does too, but it's actually using a protein primer, not like an Okazaki fragment that you may have learned about in earlier biology classes. Viral replicase will use this VPG protein, and it actually does have some nucleotides attached to it. This protein is pretty complex. It's actually a ribonucleoprotein. But it will use this viral protein to now initiate the synthesis of the antisense or antigenome and put a viral protein G on that end. These are difficult to make. Viral replicase actually struggles to make the antigenome. It only makes a few copies of it. But that same viral replicase can now make hundreds and hundreds of copies of the genome from those few copies of the antigenome. So for some reason, the viral replicase has a very easy time recognizing the VPG on this antigenome, making a copy of the antigenome. And by definition, when you make a copy of a negative strand of RNA, it is positive strand. That becomes a new genome and more messenger RNA. So by doing this viral replicase, the viral replicase is actually doing two critical jobs for the virus at the same time. It's making more messenger RNA so that more proteins can be made. But those same molecules that are being translated into protein will also slightly later become the genomes that are going to be put into the new poliovirus particles that are going to leave from that cell when that cell lyses. So that viral RNA is translated into this autoproteolytic protein, and again, I've gotten ahead of my bullets. The protease, this is a really cool point as well, and I forgot to mention this, so I'm glad I put this bullet in here. So that protease that I'm talking about as having, I'm pointing to the wrong molecule. The protease, or autoproteolytic activity, as well as being able to cleave itself into all of those 20 proteins, also has another activity. It has another specificity. It cleaves a host protein, elongation factor 4G. This is a portion of the ribosome which allows the ribosome to recognize the 5' methylguanosine cap. This is, you couldn't have designed a more evil function if you wanted to, because what this does this proteolytic activity by cleaving EIF4G makes all of the ribosomes, in essence, viral slaves. They're zombies. Because now the ribosomes inside of that cell can no longer recognize host messenger RNA, because all of the host messenger RNA has got 5' methylguanosine caps. But the viral RNA does not have those methylguanosine caps. It relies on the iris sites. The ribosomes are still functional. They can still form peptides. They just can't recognize host messenger RNA. So now all of those ribosomes can only translate poliovirus messenger RNA. They can't make any other protein. So, and if some of those proteins were going to be protective to the cell, if they were going to mount an antiviral function such as interferon, or if they were going to cause the cell to go into apoptosis, programmed cell death, those systems get shut down. And so now the cell is just becomes nothing more than a poliovirus factory. So this is a viral strategy to eliminate the translation of the host mRNA, and it's a beautifully evolved system. Just when I say beautiful, I just mean that out of respect, not admiration. This replicase generates the negative strands or the anti-genomes from the positive strand, and then many more positive strands for translation and then subsequent viral packaging. So that unusual protein that's attached at the end, that viral protein G, is attached to both the antigenome and the genome. So it's actually a promoter, as I mentioned. And it's shown here just as a protein, but in reality, that protein has got several uracils attached to it. So it's a ribonucleoprotein. And it's actually those uracils. So the protein is attached covalently, but then it will use uracils that are not shown in this cartoon as the primer for viral replicase. Here's another concern that people have from a bioterrorism point of view, is that this is a relatively small RNA genome. Technology has become such that it's not that difficult to synthesize in the laboratory RNA polymers. And people have successfully been able to just take a synthetically created base by base piece of RNA, put it into a proper cell, and out pops poliovirus. So we like to think that poliovirus is not going to be a problem for us, but there is at least the possibility that it could be used as a bioterrorism agent. And it would be tremendously effective, because here in the US, virtually no one is immune to poliovirus anymore. I mean, because the poliovirus vaccine isn't even used. People in your age have perhaps never been vaccinated. Vinny and I would be okay. This is a little bit better picture. We have a little bit of time left, of how this, these waves of proteolytic processing occur. So here's the genomic RNA as it was dumped into the cell after that uncoding process. And so that piece of messenger RNA would, using internal ribosome entry sites, be translated into this polyprotein, which would initially have enough proteolytic activity that it could cleave itself at just 2 points, here and here, generating 3 proteins, shown by these black lines in stage 1. But now, the protease, I believe it's probably here, because it's on a smaller protein, or on a smaller section of that protein, now has a slightly altered activity. It's still a protease, but now, and only now, it can recognize sites here and cleave this protein into 2. It can cleave itself here and here. It can cleave this other protein into these. So you get this second wave of proteolysis, generating a new set of proteins which have different functions themselves, including the protease. So the protease has cleaved itself yet again, and so now it has acquired the ability to cleave its remnants of itself, if you will, into all of these other proteins. So these become the final proteins. So this multiple wave strategy allows for certain functions to occur at certain times during the infection, and it ensures that the final capsid proteins, the viral proteins that form that capsid, are created late, because they will self-assemble. So you don't want to create those very early. So from one protein comes many. So it's a really highly evolved, very simple virus, but very elegant in that way. And so we have only 5 or 6 minutes left, and so it's probably not a good idea if we start in on flu viruses. So on Monday, you'll all get the flu. Have a good weekend, everybody.